Rachel Hutto is a PhD student in the biochemistry program who studies rods and cones, the cells in your eye that sense light. She studies how their signals in response to light are controlled and how this regulation is important in keeping rods and cones healthy. Please give a warm welcome to Rachel Hutto.
All right, thank you for that introduction, Jonica. So today I'll be talking to you guys about how healthy eyes work and why understanding how healthy eyes work is really important in the context of disease. But before I get into that, I want you to imagine a scenario where you look outside in your backyard and you see the following scene. It's these two cute kids and they're smiling. It looks like they were playing a game together. Adorable. So, but imagine that instead of seeing this though, you started seeing something instead that looks like this. It would be a lot more difficult to tell what was going on, right? So now you can see these kids' faces, but you don't really know what they were doing. You can't tell where they are. It's a lot harder for you to tell what's going on. Or what if maybe when you look over, you see something like this? Now you can't even make out the faces of what you're trying to focus on, of these kids. And it's really hard to tell what's going on too because your vision is kind of blurry and you have trouble differentiating color. Now, these two scenarios are not just some hypothetical thing I made up. This is a reality for people that are living with a disease called retinitis pigmentosa. And this disease is a particularly nasty disease in that it tends to hit earlier in life than a lot of visual diseases we are more familiar with. So this can affect people in childhood and usually shows up as early as age 20. And it's also a really nasty disease in that we don't have any effective treatment. We have no way to prevent this from happening. So this is a health concern that we really want to address, but it's just one of many visual diseases. And it's really a worldwide problem, vision is. So across the world, 285 million people are visually impaired in some way. And of those, 39 million people are legally blind. So this is a huge worldwide health problem. It's one we really want to address as scientists and as citizens. But before I can tell you about how we can address this problem, I want to tell you a little more about the eye itself. So this is your eye. It's not how you're used to seeing it because one, it's not in a head, and two, it's also, we're looking at it from the side. So this part of the eye that's closest to me, it's called your cornea. It's the part that you would put a contact lens on. So when light hits your eye, it actually goes through all of these parts in the front and it hits that back layer. And it's the layer that you heard about earlier, the one that I'm interested in and Allie's interested in studying, and it's called the retina. Now, this is a great drawing of it, but let's get a real picture. So here's what the retina really looks like. And what always jumps out at me when I see them is just how organized the retina is. So the retina is composed of all these different layers you heard about earlier. In an earlier talk with Allie, you guys saw, kind of learned about retinal ganglion cells and how they process signals. But the layer that I'm going to be talking to you guys about today is this one right here. And it contains the cells that first detect light and turn that into a signal. And these are called rods and cones. So rods detect signals from dim light and cones detect bright light. And there's different types of cones, so they allow you to have color vision. So the scenarios that I showed you in the beginning actually correspond to rods and cones. So that first one where you've lost your peripheral vision, you can really only see what's in the very center of your vision, is what happens when your rods start to die. And the second scenario where you've lost your ability to differentiate color, everything's kind of blurry and you can't see anything in the middle, that's what happens when your cones die. And so the big question that we want to ask, the question we want to solve, is how do we stop rods and cones from dying? But I'm not really a clinician. I'm what you call a basic scientist. So when I see this problem, I actually kind of come at it from a different direction. Instead of wondering how we can stop them from dying, I wonder what makes these work in the first place? What supports healthy rods and cones? And I want to give you a metaphor to kind of explain why I think this is important in learning about disease. So if we want to go from an unhealthy rod or cone to one that's working great, I think it's kind of like going from a chair that's been assembled incorrectly into one that you can actually sit in. So, you know, we have all seen chairs before. You guys are sitting in them right now. And so with that knowledge, you can tell from this chair on the left that the legs are just on the wrong side of the seat, right? You could easily fix this. But imagine maybe somehow you've never actually seen or maybe used a chair before. And so it would be a lot harder to figure out how you could turn this awkward object into something that you could sit on, comfortably at least. And it's kind of the same with our unhealthy rods and cones. So we know generally how rods and cones work, but there's a lot we still don't know about what's important for keeping them healthy. And if we know what's important in keeping them healthy and functioning right, we'll know what kinds of things need to be fixed when they're unhealthy and dying. So to get at that, we really want to think about the primary function of rods and cones and what's needed to support that. And as I alluded to earlier, that primary function is detecting light, and then they have to tell the rest of the retina, and eventually their brain, that they've actually seen that. So they need to detect it and then make a signal. And so the way that they do this is through these charges that are inside of them. When light hits a rod or a cone, the charges that are inside will move outside. And so you get this movement of electrical charges. And that is what creates an electrical signal. So that's the signal that, yes, this rod or cone has seen light. Now, when the light goes away, now these charges will go back in. And so if you're looking around the room or just looking at all these different lights, you can imagine that in your rods and cones, these charges are moving out and in and out and in over and over again. And it turns out that this process requires a lot of energy. Your rods and your cones and your retina in general is actually one of the most energetically demanding parts of your entire body. So we wondered, gosh, how do they meet this energy demand? What do they do? So if you're looking at any other part of your body, any other cell, the main way they get energy is through these little factories inside of them that are called mitochondria. And what these mitochondria do is they take food, like sugar, and they turn that into energy that the cell can use to do all of these different jobs that it needs to do. So we wanted to look at mitochondria inside of rods and cones. And so here's a picture of some. I'll remove the background so it's a little more obvious where the mitochondria are. And you can see they actually look kind of like that cartoon that I showed you. So these are nice, healthy mitochondria. So what we did, to kind of get an idea of how many there were, is we took a bunch of pictures, then we kind of traced over the mitochondria, and from all of these different pictures, we were able to make these 3D models of the mitochondria in rods or cones. So here's a cone, and the middle of it, where you see all those different multicolored blobs, those are the mitochondria. And as you can see, they take up a really significant portion of the amount of space in this cone. So there's a ton of them. And you know, when you see this, it's really exciting because you're like, oh, well, they have a lot of mitochondria. The mitochondria are probably making a lot of energy. That's how they get their energy needs met. Well, unfortunately, it's not quite so simple. So if you take a closer look, you'll find that the mitochondria aren't actually using much of that food to make energy. So if you take a retina and you give it a labeled sugar, so a label just so we can figure out where it goes and see if it's going into mitochondria or not, you get some really interesting results. So I'm about to show you a pie chart, and the black part of the pie chart is how much doesn't go into the mitochondria, and the blue part is how much does. So there's barely any going in there, and this is so crazy because it's so unlike almost every other cell in your body. In fact, only less than 3% of the sugar that you give a retina is actually going to go into the mitochondria to make energy. So this is so crazy, right? Like it would be if you had a city that was growing rapidly and needed more energy or more electrical power. And so the city council comes together and is like, all right, we're gonna build a new power plant, we'll invest money, time, and resources in it, go through all of the trouble of making all of these power plants, and then rarely even use them. And that doesn't make sense, right? And we have kind of weird bodies and sometimes we do strange things, but generally we don't do something that just wastes energy like that. It's not conducive to life, essentially. So that made us wonder, gosh, we have all these mitochondria, they're not really making that much energy. What are they doing? Is there something else that they're being used for, and that's why we have so many of them in our rods and cones? So we think that it actually comes back to those charges I told you about earlier. So I told you that they can move out, they can move in, but what I didn't tell you is that we found out that the mitochondria actually play a role in this, and that charges can actually go into the mitochondria and be stored there, which means that the mitochondria can actually influence how these move. And since the way these charges move is basically the signal that you've seen light, we wondered, is this process something that's important for vision? So to answer that question, we enlisted the help of two different types of animals. One is mice, and the other are these tiny little fish called zebrafish. So we wanted to look at mice because mice have a lot of rods, which makes sense. They're active at night when there's not a whole lot of light for them to see. And then we also wanted to look at fish because they have a lot of cones. And these guys are usually active during the day, so that makes sense. So first I'll tell you about some of the work we've been doing with the mice. In order for charges to get into the mitochondria, they have to go through these pores in the mitochondria. And there's kind of a set amount of them that they go through. And so when we wanted to know if this was important, we decided we'd look at a mouse that didn't have any of these. So now none of the charges can go in the mitochondria. And so we wondered, okay, how well can these mice see? Well, unfortunately, you can't just give a mouse an eye chart and ask them if they can read these letters like you would if you guys went to an eye doctor. That is not something we can do. We need another robust way to figure out how well a mouse can see. And so the way that we do that is kind of similar to a procedure you might have experienced before at a doctor's office called an electrocardiogram. So this is when the doctor wants to know kind of how well your heart is beating and if it's healthy. And so what they'll do is they'll take these electrodes that detect an electrical signal and kind of put it on your chest around the heart. And then they'll get kind of these patterns of electrical signals from your heart beating. And from the shape of that, the size of that, and all of these different attributes, they can then draw conclusions about your heart. So since there's also electrical charges moving around in your eye in response to light, we can do the same thing. You can just have electrodes kind of near your eye and then detect the electrical response to light. And so when we did this to mice that either could take up charges in their mitochondria or mice that couldn't, we saw something interesting. So if you take these rods and cones and you stimulate them with light, what you'll find is in the mice that can't take up those charges in their mitochondria, the charges leave more slowly. Which doesn't maybe sound too bad, right? What does that mean? Well, this phenomenon is called delayed recovery. And what it means is because the charges can't move in and out as quickly, you basically can't turn on and off these rods and cones as quickly. So you can't make pictures as fast. And I want to give you a little bit of intuition for what I mean by that. So first, let's simulate normal vision by watching this scene from The Matrix. So Neo is firing a bunch of shots at Agent Smith, and Agent Smith is moving really, really quickly to get out of the way and not get hit. Now, if we were watching this same scene with delayed recovery, it would be kind of like we were watching this movie on a really bad internet connection. So now we just get these still frames, and all of the fast action that's going on, we miss a lot of it. So people that have this kind of condition, if they're in a room and there's a light flickering on and off really quickly, sometimes they'll just perceive the light as always being on because they can't see that fast movement. So with that, I'm kind of want to move on and talk about some of the stuff I've been doing with the fish. So just to remind you, in order for charges to be taken up into the mitochondria, they go through these pores. And so what we wanted to know was, well, what if maybe there were more of them? So we increased the number of these pores in these fish's cones. And now they can take up more charges into their mitochondria. And we weren't really sure what to expect from this. So we knew that not having any in there could be bad, which kind of made sense to us. But if there was more, could they maybe even see better? Could they see faster? Is it a good thing? Well, it turns out it's a bad thing. So if you have too many charges in your cone mitochondria, they actually will start to break down. It's a really dramatic effect. So these are mitochondria that look healthy. You'll see they have kind of normal patterning. They're dark, which means they've got stuff inside of them. If there's too many charges, now they look like this. Yeah, not good, right? I was so surprised when I saw this for the first time. So basically, the inside of them, you can see it's just a mess, right? There's not a whole lot inside. You can see it's like whiter. And then all of these lovely patterns are gone. They've kind of just swollen up and look essentially useless. And so we thought, oh my goodness, what is happening to these fish? So we decided to look at the fish and kind of do some behavior assays. And fish are different from mice because they usually just use sight in order to get their food. Mice can rely on their sense of smell, but fish kind of have to be able to see it, especially food that doesn't move around. So here are some fish. They usually get 3 square meals a day, and today they haven't had lunch, so they're really motivated to find food. And so I give them that food, and you'll see they all go to the surface and eat it very quickly and happily. Now, if I look at fish that have too many charges in the cone mitochondria, it's a different story. So this fish hasn't eaten for the same amount of time, and when I give this fish food, she's really completely indifferent, acting like she has no idea it's even there. And so what we're trying to investigate right now is if maybe the cones with too many charges inside of them are dying, and that's why we see this decreased visual response. And if they are dying, how fast does that happen? And what does that look like? Is it similar to other diseases? And so to kind of wrap this up, I want to go back to that question that I started with, which was what's important to keeping a healthy rod or cone alive and working well? And we think we've identified a process that is important for this process of charges entering the mitochondria. Because if you don't have that process, you don't have charges inside, you get visual defects. And if you have too many, that's also bad. The mitochondria will start to break down, and you might even go blind. It might kill your cones or your rods. We also think this is really interesting in the context of disease, because in a lot of these diseases that causes rods and cones to die, people have observed that the charges are abnormal. There's either way too many, just totally in the cell, or not enough. And so it's possible that the amount of charges in the mitochondria is playing an important role in the progression of these diseases. And maybe by altering that, we could even alter the progression or maybe even slow it. So to end, by studying the healthy retina and healthy rods and cones, hopefully we can learn more about what keeps them healthy, so that we live in a world where less people see scenes like this, and more people are seeing things like this. So with that, I'd like to thank everyone in my lab that made this research possible. I'd like to thank Town Hall for hosting the event, Engage for training me. And then I'd also really like to acknowledge that my funding comes from this organization called the National Institute of Health. So if you thought that this research or research like it was interesting or important, I'd really encourage you to let your representatives know, because funding for this organization is really up on the chopping block right now. So with that, I'd like to thank all of you guys for your attention, and I'll take any questions that you may have.
Hey, good talk. And I'm not sure if I'm asking you, if I should be asking you, or if I should be asking Allison. But could you speculate? Do you think the energy needs have anything to do with why we're seeing the adaptation in the rods and cones? The firing too often might be an energy.
So it could be related. So the main energy requirement, it actually turns out that rods and cones consume the most energy when they're sitting around in the dark. And so the more they're stimulated, actually the less energy that they consume. And it's because of kind of how they have to balance those charges. So it's definitely possible that those two kind of concepts are intertwined a little bit. I don't know if there's necessarily a level of causation there, but they definitely kind of go hand in hand.
The one photograph you had there, the image, the black center, you can see anymore. Is there any relationship to macular?
Yeah, so actually, yes, it's very similar. So when you have macular degeneration, your central vision is also affected. And that's macular degeneration is a little bit different because it's not your rods and your cones that die first, But the part of your eye that's mainly affected is this part in the back called the macula. And that's actually where most of your cones are. So when that part gets damaged, you damage most of your cones, and you start seeing kind of the same thing that you saw in that image. Yeah.
Hi. My question is also about the energy usage. Does it go off when you're sleeping, or when I'm also sleeping, I guess? Or do you guys have any sort of further idea of sort of how the energy usage changes based on other things?
Yeah, so actually during the night, your eye and like different parts of your retina are consuming a ton of energy. It's actually kind of one of the most energetically demanding times for your eye. And it's kind of interesting because it seems a little counterintuitive why we would evolve like that. There's kind of different ideas for why we might do that, because generally we're active during the day, maybe we're seeing a lot of light, and it kind of makes more sense for us to only consume energy when they're totally off. But you are totally correct in that when you kind of go to bed for the night and you're no longer stimulating your rods and cones, the energy usage goes way up.
Out of curiosity,
Yeah.
is the REM state when it's most active?
Sorry, the what state?
Rapid eye movement, REM state during your sleep?
So I actually don't know anything about how maybe different phases of sleep might correlate with this energy consumption. I don't really know about that, but that's a really interesting question. Thanks.
Beautiful, and I wonder if you might be able to tell us how it is that you think the mitochondria ended up playing this secondary role of storing charges. Was it just an accident of evolution, or how did it happen?
Well, so what I didn't tell you is that mitochondria can take up charges in kind of other cells. It just seems to play maybe a really big role in rods and cones. And so why this evolved, it's kind of hard to say, but all of these charges, it's like a signal. And it's really important to control where signals are and how intense they are, because not only is this kind of a way to detect light, it can actually affect all of the different processes that are going on in the rod or cone. It can affect things like what genes are turned on even. And so if you have these things inside that can kind of control that, you get this extra layer of control of all of these different signals that could be going on, and make sure that maybe some parts of the cell see the signal and other parts don't. All right.
If no more questions, just please join me in thanking our speaker.